this is horrible. Hello. I think today we're going to talk a little bit about what goes on in Bob Wykle's lab. I'm Andy Nixon, the graduate student in the lab. We have the experienced postdoc, Dr. Johnny Wykle, and our even more experienced, highly technical tech, Diane Green. Our day usually begins by isolating human neutrophils from blood. Now, as I said before, we're a blood lab, and quite often we donate blood ourselves for our own experiments. And in order to isolate our neutrophils, we have the assistance of Diane and Johnny. Now you can see that they're in full OSHA-regulated garb so that we can protect ourselves from blood-borne pathogens. Now, I will be the donor today, and as you can see, I've donated blood before. It's usually quite painless, and I'm sure that we can find one vein that has quite not yet been totally tapped. So let me sit down. Hello, Diane.
Hello.
Now, I smoked my fair share of cigarettes last night, so my neutrophil count should be rather high. Now, this procedure is usually quite painless and not very traumatic at all for the donor. A little alcohol swab.
Nice shot.
Can we have a little zoom in there, Ben? And we usually take very large amounts of blood. Slowly, the needle goes in. The donor experiencing no pain. Ow! Usually experiencing no pain.
It didn't go in. Yes,
So-
yes, it did. Okay, go ahead.
And here now comes the blood. Our life fluid, our life force.
Hold it, John. Thank you.
And here it comes, into the tube. And there we go. Plenty of blood for a full day's worth of experiment. See? No pain whatsoever. I feel fine. Okay, now we need to work up the blood.
And we spin down our blood.
And in no time at all,
We get our neutrophil preparation. If this thing works. And here we have our neutrophils. And these cells, we use them for to study arachidonic acid metabolism as well as PAP, and to verify our working model. Here we have our working model.
darn, wrong model.
So our isolated neutrophils are used to verify this model, and we look at, or I, and together with Andy and Diane and Bob, we look at the regulation of the intracellular phospholipase A2, cytosolic phospholipase A2. And we stimulate our neutrophils with various stimuli, and we find an increase in the catalytic activity of this phospholipase A2. And the assay for phospholipase A2 normally or always ends with running thin layer chromatography. And it takes a lot of thin layer plates to do these experiments. As I said, it takes a lot of thin layer chromatography plates to get these results on the intracellular phospholipase A2. And it's a lot of scraping. But it's only 34 more plates for today, and then I can go home.
Well, Johnny, instead of doing all this work by hand, why don't you use our automated plate scraper? It's just so much more efficient and easy to use than scraping them by hand.
Great. How does it work?
Well, you put a plate on here. I have a demonstration plate. You put it on here. You set it to this 5mm setting and put on run.
Oh, nice sound.
So you collect fractions from the plate and you put them straight into the counter.
Wow, that's great, Diane. I really love the way that sounds.
It's so conducive to concentration in the lab. I'm sure you'll agree.
Now, another project we have in the lab is studying the CoA independent transacylase reaction. This enzyme actually shuttles arachidonic acid between different phospholipid species. Now, in order to better understand this, let's go to our next working model. Darn, wrong model again. Here we go. Now here is a model proposed by Dr. Wykle, or Bob as we affectionately refer to him. And we see here, at the first step, arachidonic acid is incorporated in alkeno-linked PE. This arachidonic acid can be hydrolyzed to form lyso-PE and free arachidonate. This may or may not be the enzyme that Johnny Weekender is studying in the lab. Then the most important reaction known to mankind occurs. The arachidonic acid located in alkyl PC is transferred by the action of the CoA-independent transacylase back to alkeno-linked PE. This will generate alkyl lyso-PC, or lysopath. This can be further acetylated by an acetyl-CoA lysopath acetyltransferase to form the bioactive lipid mediator platelet activating factor. Understanding this metabolism is of the utmost importance in our lab. Now often, much of our time in the lab is spent over here by our incubation bath. We will have our reactions in the incubation bath, and we usually like to have it shaken a little bit, shake things up a little. And after our reaction is done, we like to stop the metabolism by doing what is referred to as a Bly-and-Dyer extraction. This will separate the lipids from the water-soluble metabolites. It's very easy to do, usually not a problem at all. And then we take the chloroform extracts that we've recovered, and we go to thin layer chromatography, as so elegantly described by Dr. Johnny Weekender. Excuse me, a little frog in my throat.
HPLC, high pressure liquid chromatography. We take the products of phospholipid metabolism from many of the other projects in the lab, and we've isolated phospholipids in both normal and disease states. And the thing we do most frequently is make benzoate derivatives of phosphatidylcholine and phosphatidylethynolamine. When we do this, we then we subject them to a benzylation procedure, and then we put them through the HPLC after they've been properly filtered. And this is a result of a normal phase benzoate HPLC run. And here you'll see this was phosphatidylcholine. You get alkenyl, alkyl, and diacyl subclasses of phosphatidylcholine. Normal phase benzoates are injected thusly. We have the injector. Pumps are pumping the solvents through our column. Yes. Now it's going onto the column. Down, the different subclasses are isolated, and they'll come out, go into our spectrophotometer, and we can also do fraction collection if necessary after this. And this is the normal procedure for normal phase HPLC. And you have a silica column. Now, sometimes we do reverse phase. Reverse phase is reverse of normal phase. We go into the injector and back through the column. However, this does not always run as smoothly as normal phase HPLC. Oh no, look at the pressure. Oh no, I think it's going to blow up any minute.
I think we have a break in the line.
Oh no, stand back, you can get injected.
the nocturnal biochemist. He only works in the early morning hours or in the wee hours of the evening. Doug is afraid of loud noises and bright lights, so be very, very quiet as we approach Doug's domain. You see, it is only illuminated by a UV light. We need to be very quiet. I don't see Doug now. He's nowhere to be found. We may have scared him. Oh, there he is! Where's Doug?
Work in the lab with neutrophils, we have a bit of a party afterwards.
Woo, yeah! Let me have some of this Coke here. I love drinking it right over here by this bench. Yes, sir. How's it going, Junfa? What's new in the sports page?
Yeah, I read it.
Pretty interesting.
Yeah.
Quarterback has friends in PA. Well, friends are all over the place. Whew, I'm getting thirsty. Oh, hey, Ethan. How are you doing?
Yeah.
How y'all doing, guys?
Good,
Great. How
good.
about you? Want a chip?
Y'all supposed to be eating, are you?
No one really knows that we've just been, we've been doing it for quite some time now. No one said anything. We usually wait until Bob leaves.
Bob leaves, huh?
Yeah.
Yeah.
Well, I tell you what, this is not your lucky day today, guys.
Oh, no.
Oh, Osha, you're busted.
Oh, no.
OSHA?
You'll have to come with me.
Let's see the badge here.
Let's see your credentials here.
OSHA police.
Secret
What can you
agent.
do?
I'm undercover, deep undercover.
We're not eating anything in here. This really is
You have
radioactive.
chips, you have Coca-Cola,
No, that's not Coca-Cola.
you
That's
drinking coffee around.
we're doing
Coffee.
experiments. No. Those aren't potato chips either.
Drinking coffee.
No?
Coca-Cola
no,
chips?
What can you do to us anyway?
Just start out by saying, look for other jobs.
Oh no.
Out in the street?
Are you kicking us out of here?
Sorry, yes I am. I have to do my job.
Damn.
You got-
I'm never gonna get a PhD.
You should have thought about that when you're done what you did. You too, buddy. Come on. You too, buddy. Come on. Ace Ventura. Was that okay?
Has Bob left already, Johnny?
Yeah, I think he left. Yeah. You'll probably meet him downhill. This is the right time.